I also did my master's and Ph.D. in Twente, focusing mostly on immune oncology and cancer models. So I am technically a bioengineer by study, and then I focus more on biotechnology. And I was working in the past at Harvard Medical School and MIT in the US. But I'm still in contact with them on a regular basis to collaborate and work with them together. So quite a nice collaboration we have with the US here. For this lecture, a very brief outline. I added one slide more to show actually what we are doing in the group, but in general the outline will be a very brief overview of the immune system, some background in the biology, a very brief overview on what are immunotherapies, what is the definition of it, where does it come from, before we go into more the applications. We will go rather soon to cancer immunotherapies. One reason it's my background. The other reason is that most of the immunotherapies that are on the market right now are for cancer, so it's close to actually talk about it. And there are also some other immunotherapies that I will talk about, which are now more in development for the future. And then I will also talk broadly about the challenges that are awaiting the immunotherapy research. To start with our group, so we are now the engineered therapeutics department. I think when Dadang was here, it was still the targeted therapeutics. So we changed the name a little bit, but basically what we do within our group is we identify novel targets in a disease. We develop a treatment against these targets, so it's oftentimes a molecule on a specific cell type. We develop something that allows us to target this molecule. We combine it with a nanomedicine, can be a liposome, can be a polymeric nanoparticle. We evaluate the treatment in 3D models, so that was also mostly my part of the research group. And then we go to preclinical models, and then to hopefully clinical translation, oftentimes also in the form of a spin-off company. And then basically when you go to clinical translation, the whole thing starts again over and over, that you find new markers and you start the whole cycle over again. So this is what we are doing in Twente. Coming to the topic, so basically the immune system, I think it's known that the immune system is sort of the shield of your body to protect you from any infection, to protect you from germs, to protect you from bacteria. How does it look like a bit more specific? The immune system consists of a lot of different organs, which you can see here. Basically, the main involved ones are the bone marrow and the thymus, where the immune cells of the immune system originate. They can travel throughout the whole body, but what you will notice is that a lot of them are actually sort of accumulating in regions like the lymph nodes or the tonsils, which is also the reason when you're sick, when you get a cold, actually the lymph nodes are swollen, they are enlarged, which is the reason that the immune system is sort of very active in these regions. Going inside of the immune system, what we usually do is we differentiate between different barriers of the immune system or different parts of the immune system. First of all, which is technically also part of the system, are the physical and physiological barriers. For instance, your skin, your mucous membrane and nose, so in the airways, you have the cilia in your stomach. So this is the first barrier when a pathogen or a bacterium arrives. And then we have the innate immunity, which is the first line defense of the immune system. So basically when some pathogen arrives, these cells try to get rid of it right away. And then you have the adaptive immunity that sort of is the remembering this pathogen and then building up a defense once you get hit again with a cold, that your body is prepared to fight it off. So basically, this is the first line of defense. Within a couple of hours, these guys get active. And then after a couple of days, actually, these cells are more active. What I don't really like about this direct characterization is that these cell types, which we see here, which I will also discuss a bit later, are very categorized, and in reality, it's a bit more like this. So we have the innate and adaptive immune system. We have certain cell types that are really specific for the innate immune system. These are the mast cells, neutrophils, and monocytes. and certain cells which are very specific for the adaptive immunity. These are for especially T cells and B cells, but there are also some cells in between. So we have dendritic cells, macrophages, natural killer cells, which are sort of part of the innate response, but also of the adaptive response. And that's quite interesting because a lot of therapies are now also working actually on this way of dendritic cells, macrophages, and natural killer cells, and not only on the adaptive immune system. So that's important to know also for later on. When we look how does the immune system work, this is actually a very nice overview of what works. Now, this is the epithelial barrier. This is your skin. Basically, the antigen, this is the first line of defense. It cannot even penetrate. So basically, it's the easiest one because it cannot even enter your body. When it gets in, oftentimes phagocytes, like macrophages, for instance, they immediately take up the antigen and eat it up, get rid of it. Neutrophils, they kill it, or natural killer cells, they target it and kill it. That would be the first line of defense. You can also see here this takes really within the first hours after some pathogen or antigen enters the body. After a couple of days is that dendritic cells take up part of the antigen. They are presenting it to T cells, and these T cells are either communicating with B cells, which then via plasma cells actually produce antibodies against the pathogen, or these T cells change to cytotoxic T cells so they can actually target these pathogens themselves. So you can imagine if, for example, a virus like COVID, the coronavirus enters your body,